Assalamu alaikum. In this lecture, we will learn about some important MCQs relating to enzymes. So let's start. Protease enzymes are found in the many locations inside and outside of the cells. Four of these locations are listed here: lysosomes, lumen of stomach, and at a telophase spindle, and mucus and the trachea. So what of these locations are sites of intracellular hydrolysis? Means we have to find out what are the sites at which the hydrolysis process, means the breakdown of the protein, take place inside the cell. So among these, lysosomes are a telophase spindle are that particular places at which the intracellular protein digestion is takes place. So option C here is the right answer. The diagram shows a metabolic pathway. Reactant is converted into the substance X through enzyme 1, and enzyme 2 act, and the substance Y is produced, and enzyme 3 and the end product is produced. What would be the effect of adding a small amount of non-competitive inhibitor of enzyme 2? Whenever a non-competitive inhibitor will be added, then it will resist the binding of substrate with the enzyme. So in this case, the some of the active sites of the enzyme will be taken up by the non-competitive inhibitor, and as a result the amount of substance or substrate will be increased. So in enzyme 2 is act on the substance X. So if we will add the non-competitive inhibitor at that particular point, then the amount of substance X will be increased. So option B here is the rightmost answer. Two enzymes are added to a solution containing a low concentration of a substrate that they can both use. Which statement is correct? Both enzymes will use equal amount of the substrate. No, it depends upon the efficiency of the enzyme and the affinity of the enzyme with that particular substrate. Neither the enzyme will be able to use the substrate. Both enzymes will be able to use the substrate, but it depends upon the affinity of the substrate with that particular enzyme. The enzyme with the lower Pmax will use the less of the substrate than the other enzyme. No, it will use the more of the substrate than the other enzyme. So the enzyme with the lower Pmax will use the more of the substrate than the other enzyme. So option D is the rightmost answer, because it will lead to the more increase in the rate of enzyme catalase reaction. Which enzyme is extracellular? Amylase in the saliva is an enzyme that catalyzes a breakdown of starch in the mouth. Yes, as the breakdown of starch is taken place inside the mouth, so it is extracellular digestion of protein here. So ATP synthetase is an enzyme found in the mitochondria that synthesizes ATP, but it occurs inside the cells. Whenever the aerobic respiration occurs, the process occurs inside the cell. DNA polymerase is an enzyme that helps build the DNA molecules by assembling the nucleotides. It is also DNA replication process also occurs inside the cell. RNA polymerase is an enzyme involved in the process of gene transcription. So only the extracellular process is the case of option A. So option A here is the right answer. The graph shows how the concentration of the components of an enzyme catalyzed reaction changes with the time. Which line represents the enzyme with the empty active sites? Here we can say that in the case of A, all the concentration of the reactants is increasing with the time, and after a particular time it attains a steady state condition. But it is not showing any empty site of the enzyme, so it rules out option number B. As we can see here, the concentration of ingredients is decreases with the time, and then the concentration of one ingredient is showing to be get higher. It means that only the enzyme will remain free at the end of the reaction, and only we can obtain the enzyme and the products at the end of the reaction. So we can say that option B is the right answer. Which statement describes the some enzymes actions? Enzyme hold the reactant molecules so that their reactive groups are close together. And in an enzyme catalyzed reaction, more molecules have sufficient energy to react than in the absence of the enzymes, because the enzymes speed up the enzyme catalyzed reaction and lowers down the activation energy. Reaction catalyzed by the enzymes take place at a lower temperature than they would without the enzyme. Yes, if the enzyme will be present, then the lower temperature will be required to perform the reaction. So option A here is the right answer. The painkiller ibuprofen reduces the activity of an enzyme involved in the production of the prostaglandins. Ibuprofen binds reversibly to the active site of the enzyme. Which type of enzyme inhibition describes this and why? As the this particular antibiotic binds with the active site of the enzyme, so we can see that it is the competitive inhibition, right? So only the competitive inhibitor binds on the active site of the enzyme so that it can compete with that particular substrate. It will be competitive inhibition because the ibuprofen alters the shape of the enzyme. It does not alter the shape of the enzyme, it takes the place of substrate. So it is a competitive inhibition because the ibuprofen and the substrate cannot bind the same site. Yes, option B here is the rightmost answer. If it will bind with the active site, then the substrate will not be able to bind with it, and the process of pain will be reduced. Lysozymes occurs in the human tears, saliva, milk, and the mucus. Lysozymes has a role in the immune system and hydrolyzes the peptidoglycan. Which description of the lysozyme is correct? Because it is present in the saliva, milk, and the mucus and the tears, they all are extracellular things. So we can say that it is an extracellular antibacterial protein. So option C here is the rightmost answer. Which letter in the flow diagram shows the effect of adding a competitive inhibitor to an enzyme catalyzed reaction? Competitive inhibitor is a reversible, performs a reversible reaction, means it can detach from the enzyme. It affects the Vmax value of the enzyme. Yes, it does. And it affects the Km value. No, it does not affect. So, if it affects the Vmax value, then it also affects the Km value, right? So we will go towards the yes path. So if it affects the Vmax value of the enzyme, no, it does not have any effect. So option B here is the rightmost answer. The graph shows the effect of the substrate concentration on the initial rate of the enzyme catalyzed reaction. The enzyme concentration is constant, right? The substrate concentration is sufficient, but the enzyme concentration is constant or limiting. Which statement about the graph is correct? Between R and S, the number of the enzyme molecules is limiting, but between R and S, the reaction is attaining increased rate. So there is not limiting enzymes are present. Between R and S, the number of the product molecules is limiting the reaction. No, because the enzymes are producing their reaction, so there is no limiting product molecule. Between S and T, the number of the enzyme molecules is limiting the reaction. Yes, as we can see that between the S and T, this is showing a limiting amount of the reaction because the enzyme molecules has been used up by the substrate, and in other words we can say that all the enzymes have been occupied by the substrate and no more enzymes are available to catalyze a further reaction. So at that particular point the reaction attains a steady state condition. So option C here is the rightmost answer. The diagram represents the reversible interaction between the active site of an enzyme and the different inhibitors X and Y. We can see in this picture, this is the enzyme and this is its active site, and inhibitor X binds with the active sites and inhibitor binds with the other than the active site. So which row correctly identifies the type of the inhibition shown by the inhibitor X and the Y? As we can see in this picture, whatever the inhibitor binds with the active site, it is a competitive inhibitor because it competes with the substrate, right? So it is competitive inhibitor, and the inhibitor which binds with the other side of the enzyme, it is the non-competitive inhibitor. So we can say that option B here is the rightmost answer. A fixed volume of the enzyme catalase was added to a fixed volume of the hydrogen peroxide solution. The diagram shows that how the concentration of the product changes over the course of the reaction. Which explains the shape of the graph. The active site becomes more saturated. No, active sites are not become more saturated. They are using the substrate to converting into the product with the passage of time, right? The enzymes were denatured, and the enzyme is not denatured here. It is present and it's performing the reaction itself. The hydrogen peroxide inhibits the reaction. It does not inhibit the reaction. The reaction has to be performed on it. The substrate molecule were used up. Yes, we can say that all the substrate molecules has been used up and there is no more substrate present to be catalyzed. So the reaction will attain a steady state condition. So option D here is the rightmost answer. A fixed volume and the concentration of the substrate and enzymes were mixed. All other variables were kept constant. The enzyme catalyzed reaction was left until it was complete. Right? Which graph shows how the rate of the reaction changes with the time? As we can say in the option A, the rate of the reaction is increasing with the time. It does not change. And at that particular point, we can say that the the rate of the reaction attains a steady state condition, but it does not change, right? But at the particular point C, we can say that the rate of the reaction decreases with the passage of the time because all the substrate molecules has been changed into the product and there are no more substrate available to be converted into the product. So the reaction finishes at a particular point. So option C here is the rightmost answer. The graph shows how the concentration of the components 1, 2, 3 and 4 of an enzyme catalyzed reaction changes with the time. Which row identifies the components of this particular reaction? As in the case, whenever a reaction is the enzyme catalyzed reaction is performed, the most high amount of the concentration of the product is present, right? The product is present in the highest concentration. So in that particular graph, only the one line shows the highest percentage of that particular thing. So we can say that point 1 shows the product, and the point 2 shows that the line decreases and then it increases, right? So we can say that these can be the enzymes, means whenever the enzymes will be involved in the reaction, then they will be, the amount will lowers them as will be get lower as compared to the other components. But when the reaction has been completed, then all the free active sites of the enzymes will be available and the number of enzymes will be greater as compared to the other components. So point 2 shows unbounded enzymes. Component 3 means that the concentration of that component is higher at the initial point and then it lowers down. It means that it is a substrate because it is only the substrate which is used up and it has been converted into the product. So point 4 shows the enzyme substrate complex because whenever the reaction has come to an end, then the enzyme, there will be no enzyme substrate complex because all the substrate has been converted back into the product. So option D here is the rightmost answer. Which statement about the active site of the enzyme is correct? It always have a fixed specific shape. No, it can be changed with the change in the external environmental conditions. It reduces the total energy of the product. It does not reduce the energy of the product. The product has its own energy. It does not form the chemical bonds with its substrate. It forms chemical bonds. It is determined by the primary structure of the enzymes. Yes, the active site of the enzyme is determined by the primary structure of the enzyme. Lysozymes contain many different hydrolytic enzymes that may act within the cells, intracellular or outside of the cells, extracellular. Which process must occur in order for the lysosomal enzymes to do their function outside of the cell? So whenever they have to perform their function outside of the cell, they must be move out of the cell through the process of exocytosis. So option C here is the rightmost answer. The diagram shows an enzyme, its substrate and enzyme substrate complex, right? This is an enzyme, a substrate and enzyme substrate complex. Which statement explains how the substrate is able to enter the active site of the enzyme? Contact between the substrate and enzyme causes a change in the enzyme shape. Yes, it does, so that the substrate can easily bind with the active site of the enzyme. The shape of the active site and the shape of the substrate are complementary. They are not complementary to each other. They adjust themselves with each other. The substrate with the active site forms a hydrogen bonding. There is no hydrogen bonding. They are the weak chemical bonds. Whenever the enzyme substrate complex forms, the tertiary structure of the enzyme changes. It does not change. So, option A here is the rightmost answer.